Hello, I'm Katie Butts-Dehm, your Bio 110 instructor. So today what I'm going to go through is what is in your lab kit. So hopefully you've all picked up your lab kit or had it shipped to you, and you've gone through it, but just in case, when you get it, two things you want to do. First thing, you want to take out your plates. And those who are, if they're shipped, they're in your kit, your lab kit, which is in a big box. If they're not shipped, then you picked it up right out of the refrigerator. Don't leave it out in the heat. There's a side on it that says top, and very messy handwriting there. But when you put it in your fridge, make sure to put that side up, like on top. That way they're upside down, so the auger that's on the bottom will be facing the top, and water won't condense out of it, go to the top and fall back down and make a big mess. So you have it like that, where any of the water will just be staying back up in the auger. Put it in your fridge out of the way until we do that lab. Don't even open the bag or take the tape off. And hopefully there won't be any contamination, but there might be, okay? So your kit is going to come in a big box like this. And it should say Bio Lab Kit, Bio 110 Lab Kit, somewhere on it. If you have it shipped, then the box will be half that size, because of the shipping costs will make, we either have to do a shipping charge because it's $50 to ship this box, and you have like a $70 lab fee, 70 or 80. So we would end up having to either charge for shipping or go up on price or something like that. But anyway, so there it is. It's a big box. And if you had it shipped, your plates are in it. And if not, you picked them up. So there will be a list, and it should look like this. Oh, not like that. Oh, yeah, I know I'm screen sharing. Here it is. It should look like this. And if you picked it up, they were on the outside of the box. If you had them shipped, then I had to repack the kits anyway, so it should be on the inside. So your one on the inside of the box was a little bit out of date, and then it said that you get a cell phone in here. You don't get a cell phone in your lab kit, okay? So that you need to have and provide on your own, because you need to hook it to your pocket microscope. But make sure you have this list, and if it's not this exact list that's updated, then you can go in Brightspace and it'll be right there, either in Mod 1 or in the Welcome section. So let's see. Once you have the list, you're going to open the box and you're going to take things out of it. And why is this so slanted? I do not know, but there it is. So you'll have a big tub that has a bunch of stuff in it, and just take your list, and we have a small graduated transfer pipe, that's 5, and they are in this bag. And so you have inside of it lots of different things. So believe me, I know, because packing these, it's lots of packing. But here it is. You have 5 small ones that are 1 milliliter. And let's see, 1, 2, 3. Oh, there's 2 more in there. You can just take everything out of here and then put it all back in. So it'll be there. Oh yeah, there's another one. Oh, 1, 2, 3, 4, 5 smalls and 2 large graduated pipettes. And the large ones are either 5 milliliter or 3 milliliter. It doesn't make much of a difference as long as they have the little measurements on them. So we have those. Then you have a 10 milliliter graduated cylinder, which is like this. So it's, I don't have scissors. I might have scissors. Great. So it looks like that. It goes up to 10 milliliters and has markings. You should also have an either 50 milliliter or 100 milliliter graduated cylinder. Usually it's 50. I accidentally ordered 100 milliliter this time, which will work just as fine. They're just more expensive and you don't need it up to 100. What else do we have? clear plastic cups unlabeled, and there's 9. There's also 1 clear plastic cup with a line marked 1A. So there's a fill line right here. And what you're going to do in your first lab is you just fill 1A up to water, and then you measure it. So you'll pick whatever you measure it. I would use the device to measure it that's closest to that amount. Use your 100 or 50 mil graduated cylinder. And then you have these other 9 cups. And you're going to use these in place of beakers all semester. So you'll just rinse them, let them dry, and use them the next time. Because we wouldn't want to send glass beakers, and then it would increase the cost on the kits. So some kits that people have pre-prepared by lab companies are like $200 or $300. And so that's why we do it this way. It's not a lot of broken glass, and we can have them at cost. So your containers 2B and 3C that you'll use in your first lab, they're just some other kits, some other cups. It doesn't matter if they have a measurement or not on it. Let's see, then you have 3 Dixie cups. And those are probably sitting inside of your other cups, okay? Then you have, and they're not Dixie, they're just bathroom cups. So, but anyway, then you have an electronic balance or scale, and that is going to be in a box. And there's a couple of different kinds. I have mine from last semester though, so it's this kind. But you can open it, and then it has a little scale, and turn it on and turn it off, and you can turn it on and off. See if it has batteries. It may have batteries in here, which you can open them up, and then you might have to remove something to where the batteries are working, but make sure before you start the lab that it has batteries in it. We get the kits from Amazon, the kits, not the kits. We get the scales, and sometimes they have the batteries and sometimes they don't. Okay, so a known weight item A, which is a small paper clip. And if I recall, that was in here. So I probably haven't jumped out on and also a nickel. So make sure you look for that. There's a nickel. It's in here. Okay, my nickel. And if you don't have a nickel, just go get a nickel. But that's the one thing that I haven't figured out how to get reimbursed from is that nickel, because, oh, here it is, my small paper clip. So a small paper clip and a nickel, and I'm going to put them in the small weigh boats for now. Let's see, then you should have 2 large weigh boats, and these are hard to separate, so you might have 3, but 2 large weigh boats, and then you should have 4 smaller weigh boats, and these are smaller weigh boats here. So I'm going to put those back in with the nickel. Then you should have an index card that was in with that. Any index card that's a normal size index card will do, because all you're going to do is measure it with a ruler. You should have an envelope of chromatography paper and an envelope of pH paper. Now, they say chromatography paper, and it's white paper, and the pH paper is orange paper. And I used to just have them wrapped up, and I kept having people use the chromatography paper instead of the pH paper and wonder why it doesn't work. But the chromatography paper, don't touch it with your bare hands because it just might make it hurt, even though I just did it, or not make it hurt, sorry, might make it have a fingerprint in your chromatography experiment. But there it is right there, chromatography paper. If you wreck this, you can use some of the coffee filter for it. And then you have the pH paper is orange strips, okay? And it said you should have about 9. You don't really need 9, but what I would do is I would cut them in half. And then that way, if you mess up on your experiment, you still have 9 pieces of pH paper you can use. Large white coffee filters, 3 of them. So really, I know you need to use 1, but there's 3 in case you mess up your chromatography paper. You'll need to use that. Have an extra one. You might have an extra one. And then you're going to use 1 on the acid-base experiment with to strain the cabbage. Let's see, what else do we need? A wooden clothespin like this. And then you need a magnifying glass. So you might have a little one, you might have a big magnifying glass, but the thing about it is the only way you're going to need them, the only thing for is maybe looking at things in your ecosystem in a jar, or you might want to use it to, if you're identifying 5 animals and you choose some insects to identify. That might be better to figure out the species of them. So you may look at your plant leaves or something like that with them too. So I'm just going to close this now, even though the Sharpie goes back in it, but I would like to be done with it for now and get it out of my space and get this out of my space and this out of my space. Here we go. Just to let you know, those bottles of water, they are not for drinking. The bottles of water there are lab water. And I just keep refilling the bottles and use it so I can use it in here since it's not my kitchen. What else do we have? A small metric ruler. Oh, here's a small metric ruler right here. Now, this has an English side that's inches, and then it has the metric side that's centimeters, and then the really small marks are millimeters. So there's your metric ruler. A bottle each of red, blue, and yellow food coloring. So there's a small envelope. Oh, we put a lot of stuff in this giant jar since we have such a giant jar this semester. And just to make it all fit in the kit, and we throw other things. This right here is, no, let's see, here. That is food coloring, and it's also methylene blue. So make sure that you don't get the food coloring and the methylene blue stuff. Okay. So here you've got yellow, red, and blue food coloring. And then there's methylene blue, which is labeled methylene blue. I don't know why this is blurry. I think I might need to use a new webcam, but it's labeled methylene blue, literally. Let's see if I turn it here. Oh, not here. Okay, look, it's labeled methylene blue. Well, now it's just whited out. It's not blurry, but methylene blue instead of blue food coloring. So make sure that you pay attention to that. You only need a few drops of the methylene blue for the slide. So you don't need a lot of it. As far as the food coloring, since you have red, yellow, and blue, then that means you can also make green and orange if you would like. So there we go. And a lot of the things we have the food coloring for, we do need it for the osmosis diffusion lab or what was it, plant anatomy, the one where you have the celery. Okay, so you'll need it for that. But a lot of times you might just use it so you can see water better. Now we have 5 pairs of nitrile gloves. They are all medium gloves. So because my husband has huge hands and he can wear medium, and I have like just regular hands and I wear medium. So, but you might need an extra small or large. Safety glasses. And you should use the safety glasses in particular in the chemical labs, chemistry labs, but there may be 1 or 2 other that you might want to use. If you have glasses, you can wear those, but people usually put side shields on those. You can also put the safety glasses on over your glasses. Now we have our second page of the feet. And I think I printed yours two-sided, but there we have another side. So 5 paper towels of different brands. And I'll tell you, these also are hard to fold up and cut, cut the brown ones, but you have 5 different brands. And most of them say what they are, like they're printed on the paper towels already, but I did write some of them on it just so you can see. And like there's a WMGV is a Walmart grade value. So these ones you use in the second lab, the scientific method lab. Then there are 4 Ziploc sandwich bags. And the Ziploc sandwich bags, actually below it, you can see 4 paper towels rolled and held with a rubber band. So this right here is for the seed germination lab. And here's your 4 Ziploc baggies, all the same size. And then here's your 4 paper towels, which you're gonna put water on them, squeeze out the water, and fold them to where they fit in the paper towel, put them in there, and put some seeds with them. And then you have monocot seeds, which are corn seeds, and blackcot seeds, which are bean seeds. So you should have a couple of either Ziploc baggies, or I started using the seed envelopes on them. So you have one or the other, seed envelopes or Ziploc baggies. And I believe there's a few extras, more than you need for the lab, just in case some look kind of crummy. A pocket microscope that works with a cell phone. So this right here is a pocket microscope, and you're going to use this during the cell lab, and you hook it on to your cell phone, and there's a little, so before you do the lab, you might want to look, I should have some video links that show you how to use it, but you might want to practice using it with your cell phone. And it's pretty neat. It goes to from 100 to 250 magnification. I mean, I would like it if it went from 40 to 1,000. We could see the bacteria better up at 1,000, but you can see a lot of the things that we look at really good. So it's amazing that they have the pocket microscopes, I think, nowadays, because we couldn't do this lab really at home. So I already mentioned the dropper bottle of methylene blue that you use with that lab. Blank microscope slides. So it may be in a Ziploc baggie like this. Yeah, that should be. And then there's some microscope slides in there. And if you look at them, you know, I've never had any of these break in shipping or packing, even though they're just in a Post-it note and a Ziploc bag and then in a jar. But they might break now with me trying to get the tape off them. But here we have the Post-it note is new. So you have in here, should have 4 cover slips. Now this is just paper between them, and the cover slips are plastic. They used to be all glass, but nowadays they're all plastic. Little square pieces of plastic. And look, 2 of these of mine are hooked together, but now they came apart. So 4 of those. You might have an extra because sometimes they're hard to count. You should have a letter E. Oh, look, there was a one on top of that, a letter E slide. You should have a bacillus slide, which is a smear of bacteria. It may say bacteria or bacillus, and it's a large kind of bacteria. And so hopefully you'll see it. And then you should have 4 blank slides. Now I'll tell you, it's a bonus if you have another slide, because this summer we didn't do the bacteria, and I just ordered some microscopes with extra slides, and everyone got one. This is honeybee antenna, but we really needed to have the bacteria just to compare size with the other cells. Oops, sorry. So you might have an extra one. It will be a bonus if you do. You might not. I just thought, well, I'll just throw these in, and then some people have them. I wish everyone had them, but you don't need them. So there you go. Slides and cover slips. And those will be with the cell lab. And you can use the microscope. I mean, you get to keep the pocket microscope. You can take it around with you and use it outside even. Kind of neat. You'll take pictures with your cell phone and you'll turn it in. Okay, so those were all that. The large jar. So this jar is huge, okay? I think I measured it, it's about 10 inches. And this is for your terrarium in a jar. And in the spring, we have to wait till the very end to do this. And in the fall, we have to do this at the beginning. But you don't need a jar this big. You could get, like we did before, have 7-inch jars. And if you keep your terrarium for forever, they're better in glass. But you know, I'm not going to have a bunch of glass jars breaking in boxes. So we've got plastic ones, and they're good for a semester. So you'll want to set this up right toward the beginning of the semester well, the ground's not frozen and there's still plants. And so you're going to put the charcoal, take it out of the bag, put it in the bottom. Okay, and then you will put the mash and cut it to where circle where it fits in the jar. Okay, so put the mash in there and again cut it in a circle. I wonder if this size circle. No, that's a small. So I would try to cut it like maybe in the size of the jar, you know. And then put dirt on top of that, and then you'll get a piece of wood or bark from outside. You'll get a few plants. Moss is a really good plant for this, but take a little bit out of the center of a moss so it might grow back better. Maybe some little grasses or just smaller plants. Put them in there. Some people put an earthworm in there, or some of those little pill bugs or doodle bugs, whatever you call them. But they'll put those in, I think, wood lice. But so you might want to put that. You'll have decomposers from the dirt anyway, little bacterial decomposers. Then you'll water it and you will close it. And then you'll just make an observation every week. I think your lab just requires you to take a picture at the beginning and the end, but some people take them throughout. Let's see what else. Large jar, mesh, charcoal, mask. So you should have a mask and a Ziploc baggie. You can use your own mask if you want, and that's for the bacteria lab when we're gonna do the plates. And you'll breathe on one with the mask or cough on one, however you want to do that. You'll breathe and cough on another one without the mask. And that's how you'll do, and you'll compare what grows on the plate. Now, you won't see any viruses growing on the plate. So this is just for bacteria. Okay. What else do we have? The agar plates, we already mentioned them. The larger one is a split agar plate. So there's a line between it and it separates the two sides. And one side's going to be for clean hands and the other for dirty hands. So I actually was using one of my dog's paws, clean and dirty. But don't use hand sanitizer on your dog. That's poison for them and they'll lick it. So if you do that, then make sure that you just wash their paws. But you can use your own hands too. I just like theirs because it's room temp. So a lot of our bacteria is going to be body temp, but they're walking all over the floor. So let's see, what else do we have? A set of funnels. You have either red or white funnels, and there's 3 of them. Honestly, you only need one funnel, but they come in sets. What else? A bag of baking soda. So here's the baking soda, and it's in a bag, hopefully labeled baking soda. The white powder is baking soda. You use it in a couple of experiments. That's why they're such a big bag. I used to have a little dressing cup. 5 packs of yeast, which are going to be like this in a Ziploc baggie. They should all be the same kind of yeast. Well, at least 4 of them. And then balloons. So if you have a latex allergy, I don't know what you're going to want to do. Maybe not look at that experiment, get someone else's results, and have someone else like throw out your bag of balloons or something as soon as possible. You know what? I didn't see gloves in here. No, I didn't. You went to gloves. Okay. And then a large tub. And I was actually able to fit the large tubs. You might recognize these from the hospital. Maybe they gave you one of these to vomit in or to soak your limb in or your foot or hand in. I don't know. But here's a large tub. You can use your sink instead of a tub, but it is a nice way to fit everything together in the kit. Well, a Sharpie should be in there. I didn't see Sharpie written here. Maybe I skipped it, but you should have a Sharpie. You should have that every week. You should have paper towels with you every week. And some of you, no one that should probably got these jars, and you might be wondering, why are you getting these jars? Well, I want to get rid of them. No, in the beginning, the first semester, we got a bunch of the jars to use for the chromatography experiment, because normally when you're in the lab, then you have a really strong solvent like an acetone ether thing, and you have to use it in the hood, and you have to have it capped and everything like that. Well, with ours, we're just using rubbing alcohol. So, I mean, you certainly don't want to sit and breathe over it, and you can do the experiment in the jars. or mini terrariums. Who knows? You could use them for 1 week for the beakers. I don't know, but we're just using these cups, and then you'll use a pencil over it, and you'll tape your chromatography paper into it. You'll put your solvent here, and your spinach extract will be on your paper, and then it'll just like run up here and pull the spinach extract with it. But it's better to have a pencil that has sides so it won't slip off the cup. But since we're using isopropyl alcohol, rubbing alcohol, and it's at home, then I use this, but just don't breathe over it. You can use the jar though. The jars will work. And if you don't have the jars, you don't need them. You could just use this, you know? So I believe that that is it. And I just like to keep all of my stuff in the tub. And then every week, wash and rinse your cups, except this has to go back in my fridge. And, you know, and then as I use it, I might set it somewhere else. You know, certainly you'll have less and less stuff. The same thing with the weigh boats. If it's something that you're weighing that's not, you know, there are some things that you're weighing, like a nickel, that you can still use the weigh boat later on. So everything is back in here, and these and there. So this is what should be in your kit. If you have any questions, or if you're missing something, then let me know. Oh, seeds. Can't forget the seeds. I'll just put them in here. Don't have to bring any of this back unless you drop the class before ad drop, which was today. Then you have to bring the whole kit back. But otherwise, you don't have to return any of this at the end of the semester. It's paid for in your lab fee. So I guess that's it. Have a wonderful evening or day whenever you listen to this. Have a great day. Bye.